move to 1800 this evening. And so our first speaker of the day is Nicolas Rode. He's currently at the University of Montpellier, at INRAE in Montpellier, where he studies insects and specifically insects that are involved in pests. He started his education in Lyon at the École Normale Supérieure de Lyon, and then he got a PhD in Ottawa before returning to Montpellier to do important work in pest control. So, Nicolas, we're looking forward to your talk.
Thank you very much. Can you hear me well? Can you hear me okay?
Yes.
Okay, perfect. First, I would like really to thank the organizers for the opportunity to present my work today. And I thought I will give you, try to make a link between phenotypic plasticity and the first talk of the school by Laurent Loison and the microbiota. So the more of the topic of today's session and the role of the microbiota in adaptive phenotypic plasticity. So maybe first, what is phenotypic plasticity? We have phenotypic plasticity when when the same genotype have different phenotypes in different environments. So these phenotypes have different values in different environments. So let's take an example. Here you have the environment. So for example, environment 1, environment 2. So for example, this is a cold environment, this is a hot environment, and here you have a phenotype. Okay, so for example, this could be a cold environment, so let's say 10 degrees. This could be a hot environment, say 20 degrees. And then this phenotype could be body size. And in the same genotype, so if you take an isogenic line, for example, the line could have a large body size at low temperature and a small body size at high temperature. And for example, that's what we see in the pest I'm studying. It's called Drosophila suzuki. It's invasive in many countries in the world, in Europe. I think it's a little bit in Ukraine, in Odessa region. And it's also invasive in America, North America, South America, and also in Africa, in Kenya. And so what we see is that in winter, we have these really large individuals. They're also darker. And in summer, we have smaller individuals. And that's something we usually see in insects. So it's the same genotype, but that's producing different phenotypes depending on the temperature. Now, if we think about fitness, so what's the fitness function? So what's the relationship between the traits, so the phenotype, here body size, and fitness? We could think about a different fitness function. So you see here you have different curves. So this is showing how the fitness, so for example, the expected number of offspring in the next generation would change depending on body size. And in blue, you have the curve if your environment, if the environment the flies are in is at low temperature. And in that case, the fitness is maximal with this large body size. But then as body size increase, the fitness gets lower. So these smaller individuals, they would have a lower fitness in the cold environment. In the opposite, if we take the hot environment, so here in red, these phenotypes, these small-bodied phenotypes, they would have a high fitness. Okay, and the opposite here. So if you're large individuals here, you have a low fitness. Okay, so for example, this is a typical example of adaptive phenotypic plasticity. Okay, this is for body size. But today I would like to tell you more about the role of adaptive phenotypic plasticity in generalist herbivorous insects. So I told you I work with Drosophila suzuki. It's a pest and it can infest many different fruits. So for example, strawberries, raspberries, blackberries. So different fruits from more than 17 families. And our hypothesis is that these fruits, they represent different fitness optima. So that there is adaptation to these different fruits. So why do we think this? So it's a paper by Laura Lasquaga, a former PhD student, where she studied the chemical composition of different fruits. And here are it's a PCA. So basically it's a way to reduce the complexity of the composition, the nutrient composition of the fruits. And basically what you need to understand here is that we have two main axes of variation in chemical composition. And so, for example, strawberry are here. So they have a higher value on PC1, the first axis of the PCA. Cherries have a lower value here. And for example, blackberry, they differ more in PC2, the second axis of the PC. So these fruits, they differ in nutrient composition. And so what we think is because of this, these fruits, they represent different fitness optima. So here, if we have phenotype 1 and phenotype 2, this could be, for example, the ability to digest sugars, and this, for example, the ability to digest proteins, but this is just a conceptual representation. Here, it could be a fitness landscape, meaning that if you have this combination of phenotype 1, so you can digest sugar, you're quite good at digesting sugar, but you're quite poor at digesting protein, then you will perform well on strawberry. In the opposite, if you are a bit not as good to digest sugars, but you're really good at digesting protein, then you might be better off on blackberry. Okay, and as your phenotype will move away, so for example here, a phenotype that would be here would be neither good on blackberry nor on strawberry. Okay, and so our hypothesis is that flies, they need to move from one fruit to the next throughout the season, and so that they need to move from one fitness optimum to the next. And they could do this either through genetic adaptation, so by change in allele frequencies, or through adaptive phenotypic plasticity. And so the question here I want to address today is, do we see a population tracking changes in the optimum phenotype throughout the season? So for example, moving from this fitness optimum to here and to here, so with phenotypes moving throughout the season. And so this work started with Laure Lasquaga's PhD, which finished about 5 years ago. So here is France. So in Western Europe, we are in Montpellier, in south of France, and she sampled fly populations from different fruits. So either from cherries in pink, from strawberry in green, or from blackberry in black. And what she did was a reciprocal transplant experiment. So she would collect fruits, either one of these three fruits. She would, the fruits are infested, so then the fruits are in cages in the lab and the fly emerge. And then she would do 2 generations of common garden experiment, and then she would assay offspring performance, so typically egg to adult survival, in different fruit media. So the fruit media here are made either of blackberry, cherry, or strawberry. And she also looked at oviposition preference, but I will not tell you about this today. So she measured offerings performance from population that come from cherry, strawberry, or blackberry on 3 different fruit media. So here I use symbols, so squares, circles, or triangles for blackberry, cherry, or strawberry. So you see it's a kind of a 3 by 3, so you have 9 different combinations. And so I will guide you through the results. So here, the way we show this is we show performance on the original fruits. So for example, if you are blackberry on blackberry, then this would be your performance on the original fruits. And here is the performance on alternative fruits. So for example, blackberry on cherry or on strawberry. And if we see some kind of local adaptation, we expect population to perform better on the original fruits than on alternative fruits. So we expect data points to fall below this line. And so this is the data. So I will just guide you through it. So for example, if we take this data point, so you see it's in black, so the population is from blackberry, and then you either have circles or triangles, so it means that it's transfer here, it's performance on the original fruit, blackberry, and here it's the performance when transferred to cherry or to strawberry. And you can see it's below the line, meaning that the performance on blackberry is higher than performance on cherry or strawberry. And on average, the data points are below the line, so it's on average. So what we see is we see two things basically. We see a large variation depending on the fly population, and it's what we see here. So some fly population, they're better across the three fruits, and some are really bad across the three fruits. So for example, this could be due to different levels of inbreeding. And then we see this pattern of local adaptation where most of the data points, I mean, on average, these data points, they fall below the line. Okay, so we have strong variation among population, and we have a pattern of local adaptation. Sorry. And so how could we explain this? There are two main hypotheses to explain this. First, there is a genetic hypothesis that we have a local adaptation of Drosophila suzuki genotypes. So in that case, we have a genotype effect, meaning that we have, for example, outbred and inbred genotypes. That's the variation we see along the line. And then we have some genotype times and environment interaction. And that's the pattern of local adaptation we see. And the second hypothesis I want to discuss today is that the pattern we observe is actually induced by the microbiota. And in that case, it represents adaptive phenotypic plasticity, meaning that we have difference among communities, so microbial communities. For example, some yeast species that allow the larvae to develop well on the tree fruits, or the larvae, some yeast species that are poor, that provide not that many nutrients to larvae across the tree fruits. And then we also have some interaction between the microbial community and the environment. So why this? Based on this study, so it's a study published a few years ago where basically they crush strawberries and they just added agar. And here you have the time to emergence. Here you have germ-free flies. So these flies, they are germ-free, meaning that we remove, I mean, the author, they remove all the microbiota, all the yeast, all the bacteria by bleaching the embryos. And in that case, basically the larvae, they cannot develop on strawberry. So you see here a picture, they just remain teeny tiny and they never grow. On when conventional real, so with their normal microbiota, you see that they are nice and big and they have this time of emergence. Meaning that if you remove the microbiota, the larvae, they don't develop on strawberry. And so what the author did is that then they kind of added either bacteria or fungi to these germ-free larvae. And most of the time, they could restore the phenotype, meaning that they would have an emergence rate closer or just a bit different from the one of the conventional reared larvae, with only a few instances in bacteria where there was no development. And then what they did is that they also tried to isolate the compounds that would be responsible for this phenotype. So they would either add yeast, BSA, so it's kind of protein, casein, it's another type of protein from milk, or then they would use oil, lard, sucrose, glucose, or vitamins. And basically what they saw is a rescue of the phenotype when proteins were added. So yeast are really have a high protein content, or BSA or casein, meaning that the microbiota here is providing proteins to the larvae and helping them to grow. So based on this, we could have a hypothesis for the pattern we observe. So say, for example, that we have a fly emerging from blackberry, so with a blackberry microbiota. In that case, if we transfer the fly to a blackberry medium, the egg, the larvae, they can benefit from the microbiota because the microbiota grows well on this medium. And so the fly, they can use this protein, and then we see a high egg to adult survival when we have flies from blackberry acid on blackberry medium. In the opposite, if we transfer these flies to cherry medium or to strawberry medium, then the microbiota is not adapted. So the yeast and the bacteria, they don't manage to grow. And so then they don't provide proteins to the larvae, and we see a very low emergence rate. And so this microbiota-induced phenotypic plasticity could be responsible for the pattern I just told you about. So if this is to be true, we expect to see some kind of structure of the microbial community among populations of flies and among fruits. And we also expect to see some microbiota-induced phenotypic variation in Suzuki. And so that's what I want to tell you today. It's ongoing work, so of course it's like not the complete story, but we're just moving forward with this project. So we sample different fruits in Europe and in USA with many different people involved. And so we sample in France and in the USA. So we sample is a blackberry, cherry, and strawberry. And we also sample on the Réunion Island. So it's a French overseas territories here in the Indian Ocean, where we don't have blackberry or cherry there, but we were able to sample strawberry and also some tropical fruit, so mountain raspberry and strawberry guayaba. Then we had in total 50 population, and we pooled about 100 individual per population, and then we did 16S or ITS metabarcoding. So basically, we have a reference gene either for bacteria, 16S, or ITS1 for fungi, and with this, we can assay the diversity of the community of each sample. So I will jump straight to the results. See, this is an NDS and NMDS plot. So basically, it's just a way. So on average, we say that we have about 100 different microbial species in one sample. And here we're just simplifying this to put this in 2D. So here, if we have two axes, and so if we have two data points close to each other, it means that the microbial community of these two samples, they're quite similar. In the opposite, if the two samples are quite far away in 2D, it means that the communities are dissimilar. And so, for example, here, what you can see is that the blackberries are here, but they're also here. You see the cherries, they are here and here. The strawberries, they're here. The mountain raspberry, they're here. So it's a bit kind of intermingled. There is not a clear structure of the microbial community among fruits. So I would say that we have a weak isolation by ecology. So the fruits, the communities from different trees, they're not very different. In the opposite, if we look at fungi, here you have the cherries, here you have the blackberries, the strawberries, they're here. Here we have the strawberry guayaba, and here the mountain raspberry. So you see here it's much more clear cut. So it means that, and here we have samples, just a reminder that we have samples from, for example, here from France and from the USA. And they clustered together, meaning that if you sample a blackberry in France, mainland, or in the USA, their microbial communities will be closer to each other than, for example, a strawberry taken in France compared to a blackberry in France. Okay, so here we have strong isolation by ecology. So different fruits, they have different fungal communities. And then we tested the second hypothesis, meaning do we see microbiota-induced phenotypic variation? So for this, what we did is that we use natural populations. So with a natural microbiota, they were sampled on blackberry. And then we use a lab population where we remove the microbiota using bleach. And we added this microbial community to our lab population. Then we had one generation on standard medium, and then we would transfer the flies either to blackberry medium, cherry medium, or strawberry medium to assess the effect of this microbial community in 3 different environments. And so I'll jump straight to the results. So this is the jump-free control. So here it's when we don't inoculate with the microbiota. And here are the different, so we wrote the localities of the different microbial communities. So what we see is that we have different effects. Some communities, they are like bad across the board, so they would decrease performance across the three fruits, and some would increase performance across the three fruits. And the other effect we see is that some microbial communities, they are fruit specific. So if you take this microbial community here, you see the performance is high on cherry, but it's low on either blackberry or strawberry. So we have a main effect of the microbial community, the bacterial community. So it's accounting for about 17% of the variation, meaning that, so these are these main effects. So we have communities that are good across the board or bad across the board. And then we also have some interaction between the community and the fruit, and it accounts for about 5% of the phenotypic variation. Just to conclude, so we see strong variation among population, and we have a pattern of local adaptation. And now I have some funding to go a bit further to, so it's the INR, it's our national funding agency in France. And basically we want to test with these two different ways the flies could adapt to different fruits. So say, for example, that we have a switch from strawberry to cherry. These are the different genotypes. So we have alleles, like green alleles, gray alleles, or orange alleles. Okay, and what we could see is that, a hypothesis is that we have genetic adaptation. And for example, if this orange allele is selected on cherry, it will increase in frequency through time, and the mean fitness of the population will increase on cherry across through time, across generations. Sorry. And in that case, it's what we call genetic adaptation. And another hypothesis is that actually we have plastic adaptation, meaning that we don't have any change in the genotype of the host of the Drosophila larvae, but we have change in the microbial community. For example, here on strawberry, most of the larvae, they could have a green microbiota, and only some kind of a pink microbiota. And then when there is a switch, there could be a selection, but of the microbial community. So the pink microbiota increase in frequency through time. And because it's providing more protein to the larvae, we also see an increase in the mean fitness of the population. So in that case, we have actually plastic adaptation, meaning that we don't have any genetic change in the fly. It's only the microbial community that is changing through time. So that's a hypothesis for the plastic adaptation. But there could also be another way of plastic adaptation. In that case, it's just that we just have direct transfer, like so horizontal transmission of the microbiota from the fruit to the larvae. And in that case, larvae that are on strawberry, they have a green microbiota and they're okay with it. They have a good fitness. And then when the females would lay eggs on cherry, then they would have the larvae, they would have a pink microbiota, and then they would have a high fitness on cherry. And in that case, you see the mean fitness is staying the same. We still have plasticity, because if we transfer, so these larvae in this, so these larvae with a green microbiota on cherry, they would have the fitness would decrease. So we still have a pattern of local adaptation, but in that case, it's just that we have horizontal transmission of the microbiota each generation. And so that's the hypothesis we are testing now, these two different hypotheses. Okay. So just to conclude, so with this, so it's ongoing work. And so basically during the project, we studying local adaptation of the genotype. So do we see adaptation at a genetic level? And in that case, what's the underlying genetic architecture? Or is this local adaptation driven by the microbiota? And in that case, it's of plastic origin. And then, of course, it's the question we're addressing is what about the dynamics of the microbial community in the field, so on strawberry, cherry, and blackberry? Do we have transmission of the microbiota? Is it vertical transmission or is it horizontal transmission? Because the pattern we expect are different depending on the mode of transmission. And of course, we want to identify the microbial taxa that are involved in these effects, being either general effects, so microbial taxa that are maybe beneficial no matter what the fruit, and also the taxa that are fruit specific, so they are beneficial but only on one fruit, but not on another fruit. And the end goal would be to develop new approaches to manipulate, for example, the microbiota to trigger some kind of maladaptive phenotypic plasticity in the field to protect important crops, such as cherries or strawberries. With this, I'd like to thank all the people that are involved in this work, and I would like to thank you for your attention. Thank you, and I'm happy to take questions.
I'd like to start today with a question from online, if we
Yes.
have been focusing on the audience in previous days. Okay, folks online, think about if you have questions you'd like to pose. Do we have a question from the audience in person? I see one over there.
Thank you for your lecture. It's brilliant. And I wonder if can we just cultivate its microbiota and to see some metabolitic product and check if the percent of adaptation in microbiota cultivated and in your experiment the same. What type of that type of experiment can be helped, or maybe there is a problem with that?
No, it's possible. So as I told you, we can, this microbiota from Drosophila, it's cultivable. So we can isolate it on Petri dish with, some kind of selective medium. And then we can make the fly jump free. And then we can inoculate them with the microbiota we want. And so it's a way to test for this fruit-specific effect, for example. So yes, we can do that. And as you mentioned, we can go a bit further by also looking at the metabolites in the microbiota to try to get an idea, because of course these are it's always complicated to have a causal relationship. Most of the things we do are oftentimes correlative, but yes, we could try to go down to kind of pinpoint the metabolites that are involved in the phenotype we see. We think it's related to proteins, but I mean, it's, yeah, we need clearly to demonstrate this, and it's not an easy task, I would say.
Thank you.
Do we have questions online? Do we have further questions from the audience? You have,
Thank you very much for the presentation. I have a question. Since you demonstrated that most of the fitness depends on the microbiota, as for me, specifically on yeasts, why didn't go directly to the yeast infested substrates instead of manipulating with substrates? Like cut out the blueberry, blackberry, and strawberry, and isolate yeasts and grow flies on different yeasts.
Oh, okay. But thank you for your question. The thing is that first we need to know what are the yeast species that are involved in this phenotype. So, in the lab, for example, people working with Drosophila, they use Saccharomyces cerevisiae. So most of the experiment, if you tell them about yeast, they say, okay, yeah, let's assess the effect of Saccharomyces cerevisiae on the larvae. But if you go to the field and you sample larvae, you don't find Saccharomyces cerevisiae in the gut of the larvae. You find other yeast species such as Ancilospora ovarum, so some other type of yeast that are involved in wine fermentation, for example. And so it's important to first to assess the diversity. So what are the species you find on different fruits before you can assess the effect on the different fruit. You see what I mean? It's we need first to have some kind of naturalist perspective to kind of know what are the players in this to identify the species, and then we can assess their effect on the phenotype of the larvae. Does that answer your question?
Yes. Thank you for your amazing talk. I have a question about germ-free larvae. How do you obtain them?
Thank you. Yes, I was really surprised, but basically you can take Drosophila embryos and then you bleach them. So you use some kind of diluted bleach. So you know about bleach? It's a chemical we use to clean the bathroom or the the house. And so basically with this, you can remove the microbiota. I mean, you cannot remove Wolbachia. So intracellular microbiota, you cannot remove the one that are transmitted purely vertically, so within the embryo. But everything outside of the embryo, you can remove with the bleach. And then with this, you have a germ-free larvae.
There's a question.
Thank you very much. My question is very simple. So you compared the performance of flies independence of their duration of food they consume. As I understand, all those berries they share same nutrients, like they have same sugars, and you need just same types of bacteria to digest them. Why, in your opinion, there is such a huge difference in their performance? Maybe some berries contain some, I don't know, chemicals, some substances that are hazardous for mice that are not, flies that are not used to eating them. Thank you.
Yeah, thank you. So if we go back to them, so these are fruits, but here you saw here we analyze the chemical composition of the fruits, and they do differ in chemical composition. So for example, here PC1 is related to carbohydrate content, so sugar content, and PC2 to protein content, meaning that on blackberry you have a higher protein content than on cherry and strawberry, for example. So we do see differences in nutrient composition among fruits. There are fruits, but they are different from a chemical perspective. And so maybe for the larvae, it's different to be on a blackberry versus a strawberry or a cherry.
Thank you. I wanted to ask you, at the end of your experiment, when you have already induced an adaptation to the other type of fruit, have you tried to switch those flies back to their original fruit and kind of see what will happen? And if you haven't tried it, what do you think will happen? Will those flies be able to digest this fruit and thrive on it? Or they also would have to go back through this whole adaptation process?
It's a very good question. We didn't do it, but yeah, it's a very good question. And I guess it could be a way to test this. Yeah, they could. I mean, depending on how fast you lose. So, for example, if we think that when we change the fruit, we select, or we say maybe we filter the microbial community, so we only select a few species. Then depending on how strong is this selection, maybe we will lose important, some of the microbiota that is important. So for example, say that we switch from strawberry to cherry. Depending on how strong is the selection, we might lose some of the microbiota that were good on strawberry, but useless or even detrimental on cherry. And then if we go back to strawberry, maybe in that case we don't have the same fitness again. So yeah, it's a very good question, and it's related to yeah, is it symmetric? Like, could we reverse the adaptation or not? And yeah, it's a very interesting question. But yeah, we don't have a definitive answer for it.
Go ahead.
My question is based now on previous two. If different berries has different nutrients in it, and with that we can switch the berries in population. What is more fast adaptive is nutrient switch with sugar or protein difference. So if you go from blackberry to I don't know, vishnya, cherry, or you go from cherry to strawberry, what's faster? What can go faster? It's just your opinion, I guess.
So if I understand correctly, you asked me okay, maybe I will share the slide about the fitness landscape. So basically, I think you're asking about if I understand correctly, but correct me if I'm wrong, basically, you ask me what are the relative position of these fitness peaks, of this fitness optima? So which one are closer? So for example, are strawberry and cherry closer to each other compared to blackberry? In my opinion, is it what you're asking?
Yes, yes. It's
Okay. That's a good question. I mean, in the end, that's our aim is to know this. My intuition, but it's not really scientific, it's that blackberry are more different compared to cherry and strawberry. I think it's based on their chemical composition. So as I told you here, these two, they have a similar protein content, whereas blackberry have a higher protein content. Blackberry blackberries, they are in the wild. So these are wild blackberries. They're not cultivated blackberries, right? So maybe there is also more diversity in yeast on blackberry because, it's a wild, these are wild fruits. Whereas these are crops, right? But it's only an assumption. Maybe we have a reduced diversity on these crops because we have only a few varieties of cherries and strawberries. So maybe we have reduced diversity. And also if you think about the cherry orchard, the diversity of the surrounding is lower. So basically you have a lot of trees, a bit of grass, and then you have maybe a forest, but it's a bit, outside of the orchard. Whereas the blackberries, they can be in the forest, outside of the forest. So you see, I see basically blackberry as maybe something quite different even in terms of microbial composition compared to these two. But yeah, it's just a guess. Like I would need to check this basically.
Do we have any more questions? I have a question. So if I understood the first experiment correctly, you had germ-free flies, which had a dramatic loss in fitness, and then you were able to recover fitness either by adding back in bacterial communities or by adding back in fungal communities. Am I
Yes, it's just not my work, but yes, it's work from colleague in the US.
So, but either one works fine, or is there a more, if you added both back together, would that have a different effect? Bacteria and fungi, that's a very broad bacterial
Yeah.
or spectrum?
Yeah. I think that's a very good question. Do we see redundancy? Like, do we just need one yeast and we don't care about the type of yeast species, or do we have interaction? So, for example, do we have cross-feeding between different microbial species? It's a very good question. I don't work on this topic precisely, but what we know based on the literature, especially in Drosophila melanogaster, usually yeast, they tend to have an effect no matter what the other bacterial species. So a way to summarize it is that we have some good yeast and some bad yeast. And then for bacteria, it's a bit more complicated because we know that there is cross-feeding between, for example, Acetobacter and Lactobacilli, meaning that I think it's the Lactobacilli, they use, sorry, no, maybe the other way around. The Acetobacter, they use the metabolites produced by the Lactobacilli. So if we have some kind of cross-feeding. So in that case, we need the two together to be beneficial for the larvae. If you have only one, because you don't have this cross-feeding, then the larvae, they don't develop that well. But yeah, for Suzuki, we kind of just starting now, and so we still need to, describe the system and to know the species that are at play.
Yeah, and I could imagine that you have genetic variation even within species that could have an impact.
Exactly.
I have a second question. Can you very briefly describe how you would go from this understanding of microbiota and fitness respect to nutrients, and how that could actually give you a mechanism for developing a pest control system?
Wow, that's a that's a good question. We have some colleagues. So just to give you an example about what's done right now. So we have some colleagues that are so they're studying Suzuki in Sweden, and what they found is that the females, they're also attracted by yeast to oviposit because they need protein to develop the eggs basically. And right now, what they do, they published an experiment a few years ago where they would use insecticide, but at low concentration, but then they would add yeast to the insecticide. And then they would spray the leaves. I think it's with vine, so yeah, with grapes. So basically, they would spray the leaves, and what they show is that they would induce mortality with a lower concentration of insecticides because the female, they would feed on the yeast, they would ingest the insecticide, and they would die, but with a lower concentration of insecticide compared to what's normally used in this kind of for this kind of crop. So that's kind of a way to yeah, that's a way I see it. Another way, if we know that some microbiota are really fruit specific, what we could, and maybe they have a beneficial on one fruit and detrimental on another fruit, then you could spray with, a microbiota that is detrimental on that fruit, for example. And that could be a way to protect the crop. Say that you would spray cherries with a microbiota that is good for blackberries, but bad for cherries. And in that case, you would protect the cherries with this. But it's kind of a, long-term goal, right? Like I don't pretend I can do this within the next few years.
Thank you. Do we have
Thank
any?
you.
So let's thank Nicola again for not just a very interesting talk, but for provoking an interesting discussion.
Thank you very much. Yeah, have a nice day.
And we're going to reconvene at 11:30 to hear a talk from Anya Spang.